MCAT, organic chemistry. We're going to take a look at some different types of alkanol reactions, starting with dehydration, which we can call elimination. Now, with dehydration, the products formed depend on the conditions used. For example, alkenes are formed in the presence of sulfuric acid. Now, phosphoric acid is better since it does not produce as many byproducts. The correct temperature is also important because hot is used for primary, warm for secondary, and cool for tertiary. And then alcohols actually lose a water molecule in the process of dehydration. Now, with oxidation, the products depend on the type of alcohol used: primary, secondary, or tertiary. For primary and secondary, a double bond between carbon and oxygen replaces a single bond between carbon and hydroxide. But this bond will either be at the end of the carbon chain, which would make it an aldehyde, or in the middle of the carbon chain, making it a ketone. Aldehydes can be further oxidized to form carboxylic acids, and tertiary alcohols will not oxidize. Now we come to substitution, which is usually not very important for the alcohols except for halogenation using phosphorus pentachloride. So substitution may be used as an identifying test for the alcohol group. Then a reaction with active metals is just what it sounds like. When alcohols react with active metals, and in that they release hydrogen. And so this also may be used as an identifying test for alcohols. Now, esterification is a reaction between an alcohol and a carboxylic acid. And this forms an ester link, which looks like this. And this holds two carbon chains together. Now, the conditions required for this reaction are concentrated sulfuric acid and elevated temperatures. When dealing with the terminology of teeth, you may come across the terms anatomical and clinical. So we're going to take a look at what those two terms mean. So one part of the tooth is called the crown. And so the anatomical crown is covered with enamel. So the anatomical crown is all of the tooth that is covered with enamel. Then the clinical crown is the portion of the anatomical crown that is visible. So the clinical crown is the part of the tooth that you see when you look in the mouth. So the anatomical crown is a formal definition, and the clinical crown is more of an informal definition because the anatomical crown is always going to be the same. There's always going to be the same portion of the tooth covered in enamel. However, the clinical crown could change a little bit because sometimes more of the tooth is going to be showing than others. Then there's the root. The anatomical root is the portion of the tooth that is covered with cementum. And cementum is a bone-like substance that facilitates anchorage of the tooth in its bony socket. And the bony socket is called the alveoli. And then there's the clinical root, which is the portion of the anatomical root that is embedded in the jaw. So again, here you have a more formal definition. This is always going to be the same. The anatomical root is always going to be the part of the tooth covered with cementum. However, the clinical root could change because in a patient with advanced bone loss, the clinical root may be reduced in size. And then also, someone may have a receding gum line. So when someone's younger, the clinical crown might be kind of small, but as they get older and the gum line recedes, then the clinical crown is going to become bigger. However, the anatomical crown is going to be the same because it doesn't matter how much of the tooth is showing. And then we have the cervical line, which separates the anatomic crown from the anatomic root. Notice the cervical line doesn't have anything to do with the clinical crown and the clinical root. That's because that could change some, whereas the cervical line is always going to be the same because the anatomic crown and the anatomic root are always going to be the same. Now, the cervical line is basically the junction between these two tissues, the enamel and the cementum. So another name for the cervical line is the cemento-enamel junction because it's where the cementum and enamel join together. So an abbreviation for the cemento-enamel junction would be the CEJ. And then another name for this area is called the cervix of the tooth, and that is spelled CERVIX. So that's a look at the anatomical and clinical parts of teeth.
Antibodies. Antibodies are also known as immunoglobulins. And they are large Y-shaped proteins produced by B cells. Now, an antibody is used by the immune system. You might have been clued in by the immunoglobulin alternative name. So it's used by the immune system to identify and neutralize foreign objects such as viruses and bacteria. So if you inhale or ingest something that is foreign to your system, antibodies are going to kick in and try to neutralize these foreign objects. The antibody recognizes a unique part of the foreign object called an antigen. So it's important to remember the difference between these two, since antigen and antibody are both similar to each other. So the antigen is what's part of the immune system and is going to stop the foreign objects. The antigen is a specific part of that foreign object that we're going to try to neutralize. Each tip of the Y, remember we said an antibody is a Y-shaped protein. Each tip of the Y has a paratope. And think of the structure of a lock for a paratope. And each paratope will only bind to a specific epitope. And think of it like a key. So let's look at our structure over here. We've got our antibody, which is this whole Y-shaped protein, and then we've got all these antigens up here. So there are lots of different antigens that could come into your body, and this is probably not what they look like, but it's just to give you an example. If we're thinking of our antigens, and this area here is going to be our epitope, this part here that would bind, it's supposed to be considered a lock. So if this, it's supposed to be considered our key. So if this is our lock here, this antigen binding site, or the paratope, then it's only going to fit with one key. A lock is only going to be unlocked with one key. You have to have the right key to get it to unlock. So we've got our antibody, and each part of the Y is only going to bind to a specific antigen. The paratope, this little shape on the end of our antibody, is only going to bind to one epitope, which is located on an antigen. So that one spot on your antigen is what's going to be able to bind to the one spot on the antibody. And if you don't have the right key to go in the lock, or the right lock to bind with the key, then nothing's going to happen. So there are lots of antigens, and different antibodies can attach to different ones of those. But one antibody is only going to be able to bind to one antigen. If you have lots of antibodies floating around in your body, each one is going to only be able to attach to one antigen. So you may have lots of them that can attach to one antigen and lots that can attach to another, but you can't just say, okay, well, I don't really have this antigen in my body, I have this one, so let me plug that in there, because it's not going to fit. You have to think of it like a lock and key. So once you have this antibody produced, it's only going to be able to bind to the one antigen to neutralize it. So once the antigen and antibody bind, and remember, special epitope-paratope relationship, lock and key, it's only going to be able to bind to one specific type of antigen. Once they bind, the foreign object is tagged for attack by another part of the immune system, or it's neutralized directly. There are some antigens, some foreign objects where if we block the antigen, then that foreign object isn't going to be able to do any more harm in your body. So the antibody can neutralize it directly. But there are other foreign objects that can't be neutralized quite so easily. So instead, there is something secreted by the antibody, and it coats the foreign object, and then the foreign object is tagged for attack, and some other part of the immune system steps in and removes the foreign object. So now that you've seen your antibody and seen how it binds to antigens and how it can either get rid of or neutralize the foreign objects directly or alert the attack for another part of the immune system, let's go back to the B cells. Remember, antibodies are produced by B cells. So once a B cell has been activated, once there's some foreign object in your body that the B cells know about and they say, okay, we've got to do something now, we've got to get rid of this. The B cells can turn into antibody producing cells called plasma cells. So they can produce new antibodies, these plasma cells that the B cells turned into, or they can be turned into memory cells that can remember an antigen and respond faster in the case of future exposure. So these B cells can turn into plasma cells that produce more antibodies if needed, or they turn into memory cells. And the memory cells are going to remember the last antigen they had, and they're going to be able to respond faster. So once this antibody is attached to this antigen, it's going to remember it next time, and it's going to be able to respond faster. It won't have to try all of these keys to figure out which one goes in the lock. It will already know which key goes in the lock, and it will be able to respond faster. So that's why if you've been sick with something before, you might not get it again, like chicken pox. Once you've had it, you usually don't get it again. Let's see. You get a flu shot and it's got antibodies in it. Once your body learns how to attack that flu virus, at least that particular strain of flu virus, then it will be able to attack it if it comes into your body again. Now, different strains of flu virus are going to have different antigens, so it won't be able to attack every flu virus, only the ones that have that same antigen that your flu vaccine had in it. So antibodies are very helpful with maintaining your immune system and keeping you healthy, or keeping any organism healthy. And the B cells produce the antibodies. The antibodies find foreign objects, find the antigen on them, and the particular epitope that it can bind to. And once they bind, that foreign object is tagged for removal. It's either going to be attacked by another part of the immune system, or it's going to be neutralized directly, and it won't be able to harm anything and will eventually get transferred out of the body. And you've got your plasma cells that are going to produce more antibodies for your body, and your memory cells that are going to remember past antigens, past foreign objects that have come into your body, and know how to respond to them faster in the future. So remember, antibodies are also called immunoglobulins, and that will help you remember it's a part of the immune system. Remember the difference between antibody and antigen. The antibody is what is fighting the foreign object. The antigen is that specific part on the foreign object that has to be bound to be able to attack that foreign object and get rid of it. DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA, the DNA in all three would be the same, and it would let whoever was looking at this know that all three of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curved shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine thymine pair or guanine cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form 2 long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Enzymes. Enzymes are highly selective catalysts that greatly accelerate the rate of reactions. So they speed up how quickly a reaction takes place. Most enzymes are proteins, but some catalytic RNA molecules have been discovered. So there are RNA molecules that will catalyze reactions and help speed them up in the same way that these proteins will. And so they both work as enzymes. In enzymatic reactions, the molecules at the beginning of the process are known as substrates. During the reaction, these substrates are turned into different molecules known as products. And at the end of the reaction, the enzyme will be unchanged. The enzyme may have to change shape a little bit to bind to the substrate and finish the reaction, but the enzyme itself will be unchanged. It will not have turned into different products. It will not have gotten bigger or smaller. It will remain unchanged. Enzymes work by lowering the activation energy of a reaction, drastically reducing the reaction time. If you had to build up all that energy to complete a reaction normally, it would take a really long time. Well, whenever the enzyme works on a reaction, it lowers the activation energy and makes the reaction happen a lot faster. In fact, a lot of our metabolic processes would not be fast enough to sustain life without enzymes there to speed up the reactions. Enzymes are highly specific for their substrates. That means that each enzyme will only bind with specific substrates that fit with its active site. So each enzyme has an active site. Here we've got our enzyme. To give you an example, this area would be the active site. So both here and here, this open area in the overall shape of the enzyme is the active site, and each enzyme has an active site, and only substrates with a similar shape that would be able to fit in there and bind without the enzyme changing shape too much will be able to fit with that enzyme and complete a reaction. So this would be your substrate. And this is where the substrate would be coming into contact with the enzyme. Here you can see the enzyme and substrate have bonded, and now they have formed the enzyme-substrate complex. And you can see that the enzyme changed shape a little bit to bind to that substrate. And then in this next phase you have the enzyme product complex, where the substrate has started to turn into two different molecules and separate from each other. And then in this last part, you've got your enzyme remaining unchanged, and the products, two separate molecules that the substrate has been turned into, will leave the enzyme. So you can see the enzyme remained unchanged, and the substrate was turned into two separate products. And going through this stage is going to lower the activation energy of a reaction. Enzymes can be affected by inhibitors, which are molecules that decrease enzyme activity, and they can be affected by activators, which are molecules that increase enzyme activity. So where you have enzymes that are raising the rate of speed of a reaction or lowering the activation energy needed, the inhibitors are going to slow that process down, and the activators are going to speed that process up. So where the enzyme is acting on a chemical reaction, these other molecules can also interfere with the chemical reaction and either speed it up or slow it down. The main thing to remember about enzymes is that these are highly selective catalysts, and they are going to increase the rate of a reaction by lowering the activation energy needed, and at the end they are going to remain unchanged. Genes. A gene is a molecular unit of heredity of a living organism. A gene is the basic instruction, a sequence of nucleic acids. So a gene is going to be the instruction, it's going to be the sequence of nucleic acids that tells the cell to code for a certain protein or for a certain RNA chain. It isn't just one little thing, it's a sequence that gives an instruction to the cell. While an allele codes for one variant of that gene. So if you've heard someone say, oh, she has really good hair genes, or she's got the gene for blue eyes, that's not exactly true, because everyone has a gene for eye color or hair color, and for every other trait that you have. But what determines what color your hair is or what color your eyes are is going to be the alleles for that gene. So there are dominant alleles and recessive alleles. And if you have even one dominant allele, then you're going to have the dominant gene. If you have two recessive alleles, then you will have that recessive gene. For instance, if you have two parents that have brown eyes, you could still end up with blue eyes because blue is a recessive gene. So both of your parents had one dominant and one recessive brown and blue eye color allele. The brown would have been prevalent in both of them. But since they both had that recessive blue allele present, you could still end up with blue eyes because of the recessive alleles combining. And I know that all sounds a little confusing, but it's just to tell you that each gene doesn't determine something all on its own. It's the alleles that are the variants of that gene that determine what the outcome of each gene sequence is. Genes do specify all proteins and functional chains. So they specify how these are going to be created, the functional RNA chains. Genes hold the information needed to build and maintain an organism's cells and pass genetic traits to offspring. So where I was talking about two parents that could have brown eyes and still pass on the blue eye color, it's because of the alleles that they carry, whether dominant or recessive, whether you can see it as a physical trait or not. Your parents carried genetic traits and they passed those on to you. And some of your physical traits may not all be the same as one or both of your parents, but the genetic traits that you got did come from the alleles that they had combined and mixed together. So you took some of your mother's alleles and some of your father's alleles, and when those combined, you ended up with different alleles and different genetic traits of your own, but they both came from the genetic traits your parents had. Some examples are eye color, number of limbs, and blood type. So some genes you can see right away manifested, such as eye color or number of limbs. But blood type is something that you would have to do a little more scientific research to discover. You can also search for certain markers in your body for diseases such as Alzheimer's or other hereditary diseases. Those can be tested for, and your DNA will show, your genes will show if you have a likelihood of getting that disease. And so that's definitely not something you can see just by looking at someone. It's something that you would have to do more research into. But it's possible by looking at the genes in your body. Now, for a gene to create or code for its protein, it must go through transcription. And transcription is where single-stranded messenger RNA or mRNA is created that is complementary to the DNA from which it was transcribed. And we'll look at that over here in just a minute. But basically, you're starting with DNA and you're transcribing it into the messenger RNA. And then the messenger RNA is going to bring the message elsewhere in the cell. So next comes translation. That messenger RNA is used as a template for synthesizing a new protein. Transfer RNA, or tRNA, brings the amino acids and anticodon necessary for protein synthesis. And something to remember is that genes are read 3 nucleotides at a time in units called codons. So you look at the 3 nucleotides that are coding for this one protein, and they're going to be transcribed onto messenger RNA, which is going to travel elsewhere in the cell, and transfer RNA is going to bring the amino acids and the anticodon that match up with that set of 3, with that codon, and let the protein be created. So let's look at that over here. Let's pretend this gene sequence came from a strand of DNA. And remember, your bases are adenine, guanine, cytosine, and thymine. So we are going to say it's going to change from DNA to the messenger RNA. So we're transcribing here, and each base must be transcribed. So here we've got our transcription. So the guanine is going to transcribe to cytosine, cytosine to guanine, thymine to adenine, cytosine to guanine. There we go. Thymine to adenine, and thymine to adenine. So the messenger RNA is going to carry this sequence. So you can see how it isn't exactly what was on the DNA. It's what base would have paired with the DNA. So it's kind of like a code you've got to sit here and figure out. You've got to decode what the messenger RNA says. So the messenger RNA has the pairs that would have gone with the DNA, and so in comes your transfer RNA, which is going to have its amino acids and the anticodon that's necessary for each of these. So each set of 3 is a codon, and it gets transferred over. And so now the transfer RNA is going to come in, and it's going to bring what we need. So it's going to say, okay, we had cytosine, so now we need the guanine. I guess we should really point this way. And then it's going to bring cytosine back to pair with the guanine, thymine back to pair with adenine, cytosine back to pair with guanine, thymine to pair with adenine, and thymine to pair with adenine. So now in a different part of the cell, you've got these codons, and they're gonna code for a certain protein. And this is what happens in the cell. So this part would be your translation. So you've got strands of DNA. Part of the cell says, oh, I need a certain protein. Well, this codon codes for one of the proteins you need, and this codon codes for one of the other proteins you need. So the DNA is going to send it. The messenger RNA is going to hold the transcription, which is where it's changed to what the pairs would be. What pairs with each base is going to be the transcription that goes on the messenger RNA. And then the messenger RNA travels off into the cell to where the protein is needed. The transfer RNA is going to bring the bases plus amino acids. And then you're going to get these bases plus the new amino acids are going to give you your proteins that these codons were coding for. So you've got your transcription where it's coded onto the messenger RNA, and then you've got translation where it's decoded and the transfer RNA brings in what you need plus amino acids and your proteins are created. So that's kind of a complicated process, but it's what genes do. That's their job. Their job is to send messages throughout the cell so that new proteins can be created. And they are going to carry your hereditary information, and they have the basic instructions necessary to tell your body how to carry out the hereditary processes and keep producing the genes that keep producing the cells and the proteins that you need and maintaining your cells. So just keep in mind, genes are units of hereditary information, and they carry messages throughout the cell and instruct the cell on how to create the proteins that your body needs. Kingdom Animalia. Animals have several characteristics that set them apart from other living things. Animals are eukaryotic and multicellular. So where other kingdoms can have creatures that are comprised of just one cell, animals are going to have more than one cell. They are also heterotrophic. This means they cannot create their own food and must rely on other sources of nourishment. So they're going to eat other plants or animals. They're not going to be able to create their own food with sunlight like plants can. Animals do not have cell walls, but they may have an exoskeleton, which is like a hard skeleton they secrete onto the outside of their body, or a shell. Now, they don't have to have that. Obviously, we don't have a hard exoskeleton or a shell, but some animals like your crawfish or lobsters would have an exoskeleton, or things like snails have a shell. All animals are motile. This means they can move, even if only during certain stages of life. So some animals may only move a little bit, or very slowly, but all animals are motile, even if it's just for a short period of time or during one stage of their life. Nearly all animals undergo some form of sexual reproduction, but some animals are also capable of asexual reproduction. And this is possibly, and a lot of times, through parthenogenesis, which is when a fertilized egg is produced without any mating, budding, or fragmentation. So it is possible, but most of the time the animals are going to undergo sexual reproduction to produce offspring. Animalia is also known as metazoa, as far as a kingdom name goes, and it is divided into the subkingdoms, Parazoa, which consist of sponges and trichoplax. Sponges, I'm sure you can think of, but trichoplax kind of looks like a puddle you spilled on the floor. It's very flat, but it is still an animal. It does have differentiated cells. It even has some eye cells on it. So Parazoa, such as the sponges and trichoplax, have differentiated cells but no distinct tissue or tissues or body symmetry. So they're kind of oddly shaped, where most animals you would be able to see some symmetry. There is no symmetry as far as Parazoa are concerned. Now, all other animals are put into the subkingdom Eumetazoa. And these have 2 or 3 distinct layers of cells with differentiated tissues and either radial or bilateral symmetry. And what that means is that you can divide them a certain way. If they have radial symmetry, you would be able to divide them any way going in a circle, and the halves would match up. So a jellyfish would be an example of that. If you divided the jellyfish here, if you could divide it across here, if you could divide it across here, going any way in a circle around the top of the jellyfish, down across the top and down through all the tentacles, if you cut it, it would be more of a circular shape, and anywhere you cut it, it would be the same on either side. So it's symmetrical. And it would have radial symmetry because there isn't just one symmetrical line you can cut along. There's not just one line of symmetry, there are several. The same goes with a starfish. You could cut it along a line going this way, 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 or a line going this way. If you go all along that circle, it can have symmetry. It can have all these different lines of symmetry, and what's on the other side of the line of symmetry is going to match what's on the first side of the line of symmetry. Bilateral symmetry, you can only divide the body in half one way. So people have bilateral symmetry. You can divide straight down from the top of your head to between your 2 feet, and the 2 halves should match up pretty well, at least as far as body type and tissues are concerned. So people have bilateral symmetry. You can divide straight down the middle and the halves should match up pretty well. Butterflies or moths are another example that should match up. Cats, dogs, a lot of animals display bilateral body symmetry, but there are some that have radial symmetry. But these are all Eumetazoa. Remember, the Parazoa is not going to have any kind of body symmetry because it doesn't have distinct tissues, and it is not going to have a distinct body shape every time. It's going to be different. So whenever you are thinking about kingdom Animalia, remember these certain characteristics that set them apart. They're eukaryotic and multicellular, made of more than one cell. They are heterotrophic. They cannot make their own food and must receive nourishment from other sources. They do not have cell walls but may have an exoskeleton or a shell. And all animals are motile. They are able to move at least at some stage in their life. Kingdom Fungi. Fungi are a group of eukaryotic organisms. They used to be classified as plants, But genetic studies show that fungi are actually more similar to animals. Now, one reason that fungi were originally classified with plants is because they have a cell wall, just like plants do. But fungi have cell walls made of chitin, while plant cell walls are made of cellulose. So one of the differences that set fungi apart from plants is that their cell wall was composed of something completely different. Fungi can reproduce asexually via vegetative spores, or they can reproduce sexually via meiosis. A fruit body, such as a mushroom or mold, is formed where spores are created and released to begin new colonies. So when you think about a plant and how it produces a fruit, and that's where the seeds are that carries on life, it's kind of the same thing, except fungi and plants are not the same kingdom. So a fungi's fruit body is going to be in the form of a mushroom or mold, and instead of having seeds, it's going to produce spores that are released, and the spores go out and they create new colonies of fungi. Fungi include yeasts, molds, rusts, and familiar mushrooms. Something to remember is that slime molds and water molds, even though they are molds, are not grouped with true fungi. They used to be, but they are no longer considered true fungi because they just don't fit into this kingdom. So even if you see the word mold, that doesn't mean that it's definitely something that fits into kingdom fungi. Fungi grow in soil, on dead matter, and is symbionts of plants, animals, or other fungi. And being a symbiont means that a fungus can live on a plant, on an animal, or on some other fungus. So it can live on it. It could be a neutral relationship where that fungus lives there and it doesn't affect the plant, animal, or fungus that it's living on. It could be something positive where they're both getting something out of it, or it could be something negative where the fungus growing on that plant, animal, or other fungus is causing a problem. For instance, on certain rice plants, if a fungus grows on it, it can cause a disease that causes all those crops to go bad and not be good for consumption anymore. So it can be a bad relationship, or it can just be something neutral, or it can be something that's helpful for both the plant, animal, or fungus and the fungus that is growing on one of those things. And then a fungus doesn't always have to be a symbiont. It could just grow in soil or on dead matter all by itself. Fungi uses can include an essential role in decomposition. So when fungi grow on dead matter, they're going to help decompose that dead matter. It can be a direct source of food. Familiar mushrooms would be direct sources of food that you could eat, though not every mushroom, because fungi can be toxic. So don't think that every mushroom you see is all right or every fungus is going to be helpful. Fungi can be used as a leavening agent for bread. That's a yeast. Fermentation, such as in wine, beer, and soy sauce, and that would be yeast again. Antibiotics, enzymes and detergents, biological pesticides that could kill certain weeds, and then remember, fungi has a lot of uses, but it can be toxic. So kingdom fungi is going to consist of eukaryotic organisms, and they're going to have cell walls made of chitin. They can reproduce asexually or sexually, and they are going to include yeast, molds, rust, and familiar mushrooms. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have one copy. So, in this first part, this would be interphase. And it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase. And the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy. And they're going to cross over a little bit at these points here. And where those cross over, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair. And that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And you can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a two-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become two distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those two cells. So now there are two diploid daughter cells. So we had our diploid germ cell. The DNA, the genetic material was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis 2, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the two daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now, and we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite sides still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitochondria. When you see the word mitochondria, you're looking at the plural form of the word. The singular form is mitochondrion. So whenever you see those, don't be confused or think you're looking at a different word. It's just the singular and plural forms. First, let's talk about the structure of mitochondria, and then we'll talk about its functions. So the structure of a mitochondria. If you see it floating around in the cytoplasm, it kind of looks like a jelly bean. If you can see a cross cutout of it, then you're going to see this little squiggly membrane going through the middle there. So let's look at what each part of the mitochondria is. This outer layer that you can see is called the outer membrane. Next, this space in here between the outer membrane and this next line is called the intermembrane space. And then this next line is the inner membrane. So that makes sense, right? You've got your outer membrane, your inner membrane, and then the space between it is just the intermembrane space. Outer, inner membrane, intermembrane space, the space between the two. Next, you've got the space created through all these little zigzaggy patterns of the inner membrane, and the spaces that are created here and in there and down in here between where it zigs and zags, and you've got that space between each little fold. That is called the cristae. And then, last but not least, everything inside that inner membrane is called the matrix. So there you've got your structure of a mitochondrion, the basic parts of it, and it's important to know that they are found in nearly all eukaryotic cells. So that's not all, but most eukaryotic cells, and that's going to be your larger cells, the ones you find in plants and animals. And within the cell, it's going to be found in the cytoplasm. So you will see mitochondria floating around in the cytoplasm. And you could have one or you could have lots of mitochondria. It kind of depends on how big the cell is. But the basic structure is going to be outer membrane, intermembrane space, inner membrane surrounding the matrix with the cristae forming in the folds between that inner membrane. Okay, let's look at the functions. Mitochondria produce adenosine triphosphate, which is known as ATP. So if you see ATP, it's an energy source and it's called adenosine triphosphate. But A lot of times this is not going to get all typed out. You're just going to see ATP because it's a very common abbreviation in biology. Now, ATP is produced through cellular respiration, which is also known as aerobic respiration. And that's because it involves oxygen. It's basically the cell's breathing cycle where it gets to take in oxygen and use it. So aerobic respiration or cellular respiration are basically the same thing. It just whoever's typing or writing the paper or information you're looking at may use cellular or aerobic, but basically it's going to refer to the cell taking in oxygen. Now, the set of reactions that produces ATP is known as the Krebs cycle. So, the mitochondria is involved in the Krebs cycle because it goes through some of those reactions to produce ATP. And the mitochondria is sometimes called cellular power plants or powerhouses because they create the cell's primary energy source, which is ATP. So since they create the primary energy source that the cell has to use, they are called cellular power plants. They also can regulate the cell's metabolism, and they are involved in cell division, cell growth, and cell death. So all of these processes require energy, and since the mitochondria is producing the primary source of energy, it makes sense that the mitochondria would be involved in a lot of these processes. It has a lot of important jobs, but the most important is going to be producing ATP because that is the primary source of energy for the cell, and the mitochondria is what produces it. So it's very important to remember production of ATP, power plant of the cell, for mitochondria because that is what you're mainly going to see it associated with. Now it can also do these other things, regulating metabolism, being involved in cell division, growth, and death, but main thing to remember is producing ATP. And you've got your structure, so remember outer membrane, intermembrane space, the space between the two membranes, inner membrane, cristae between those folds, and the matrix. Remember that mitochondria are going to be found in eukaryotic cells, and they're found in almost all eukaryotic cells, but not every single one. And most eukaryotic cells will have at least one, but they could have a lot of mitochondria, and you're going to find them in the cytoplasm. So just remember, if you're looking at a cell diagram, you're looking for that little jelly bean shape, and if you're thinking about what does a mitochondria do, think about producing energy, producing ATP. The mitochondria is your power plant of the cell. Mitosis. Eukaryotic cells reproduce asexually by dividing into two genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So, this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, First stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Nucleic acids. Nucleic acids consist of a linear chain of nucleotides. So that leads us to ask, what is a nucleotide? Well, a nucleotide is going to consist of a nitrogenous base, which could be a purine, such as adenine, which is usually abbreviated with A, and guanine, which is usually abbreviated with G. Another nitrogenous base is a pyrimidine. Another type of nitrogenous base is a pyrimidine, and those would be cytosine, thymine, and uracil. After you get your nitrogenous bases, you also need a pentose, which is a 5-carbon sugar. The most common ones you're going to see are ribose and deoxyribose. And once you have your nitrogenous base in your pentose group, you need a phosphate group. And when you put all three of these together, you get your nucleotides. So let's look at some common nucleic acids. First, we have ribonucleic acid, or RNA, which consists of the sugar, ribose, so our pentose, our 5-carbon sugar ribose, plus adenine, guanine, cytosine, and uracil. Those would be the nitrogenous bases that you're going to use. And ribonucleic acid, or RNA, is usually single-stranded. The next one would be deoxyribonucleic acid, or DNA, and you notice that Na in both of them is there. It stands for the nucleic acid part. The first letter is telling you which sugar it's going with, which pentose. So deoxyribonucleic acid is going to be with the sugar deoxyribose, plus adenine, guanine, cytosine, and thymine. And you might notice that RNA is going to use uracil as its second pyrimidine, where DNA is going to use thymine as its second pyrimidine. And that's how it always is. RNA is going to use uracil, DNA is going to use thymine. So you need to remember that part. DNA can be single or double-stranded. It is a bit more advanced or complex than the RNA, so a lot of times it is double-stranded, but it can be single-stranded as well, where RNA is usually going to be single-stranded and only rarely double-stranded. Something to remember as well is that adenine always pairs with thymine or uracil, and guanine always pairs with cytosine. And the way I like to remember that is that the curved letters, guanine and cytosine, our abbreviated letters, go together. And then our letter abbreviations that are either made with straight lines, like A and T, go together, or vowels go together. But if you just remember that the curved letters go together, you can always pair up G and C and then pair up whatever's left. And it will be adenine and thymine in DNA and adenine and uracil in RNA. Genetic information is carried by DNA in all organisms and most viruses. However, some RNA, some viruses use RNA. So, most viruses will use DNA for their genetic material, but some viruses will use RNA. So you can see that nucleic acids are very important for cells. They're made up of a nitrogenous base or series of bases that are going to pair together the same way every time, pentose or sugar, and a phosphate group. And once you get your nucleotide chain and it forms its nucleic acid, you can name it based on which sugar is joined with the rest of it. So ribose formed ribonucleic acid, deoxyribose formed deoxyribonucleic acid, and the nucleic acid's most important function is carrying genetic material. It's going to be carrying the genetic material for all cells and organisms. Photosynthesis. Photosynthesis is a process used by plants and other autotrophic organisms to convert light energy, usually from the sun, into chemical energy that can be used by the organism. Photosynthesis usually gives off oxygen as a waste product as well. Oxygen is not something that the plant or other autotrophic organism will need in most cases, and so it will give off oxygen as a waste product. Now, autotrophic means that these organisms make their own food. They are capable of creating their own food. They can harness the light energy and create their own source of nourishment. Heterotrophs cannot do that and must rely on autotrophs or other heterotrophs for their nourishment. Most plants, most algae, and cyanobacteria perform photosynthesis and are called photoautotrophs. So, autotrophs are organisms that can create their own food. Photoautotrophs are organisms that create their own food from light, since photo translates to light, looking at the Latin root for that word. These are organisms that can create their own nourishment from light. Photosynthesis maintains atmospheric oxygen levels and supplies most of the energy necessary for life on Earth. So, as these autotrophic organisms take in carbon dioxide, they let out oxygen and help maintain the atmospheric oxygen levels, because people and other organisms exhale carbon dioxide, and the plants or other autotrophs take that carbon dioxide in and turn it back into oxygen, which we breathe. So if we didn't have photosynthesis, we'd be running low on oxygen. And so photosynthesis helps maintain our oxygen levels. It also supplies most of the energy necessary for life on Earth. Even though you don't feel like you're eating light or getting your energy just from light, if you eat a vegetable, then you're eating something that it has only been able to grow because it used light energy to provide nourishment for itself. So you are now eating that plant, which got its energy and was able to grow from light. If you were to eat a cow, if you had beef, if you had a hamburger, then you would be eating another heterotroph. Cows can't make their own food, but they ate grass and other vegetables and other foods that were grown, and they were grown by having light energy harnessed inside those plants. And so the cow got the energy from the plant, which got the energy from the light, and then you were getting energy from the cow. So it all comes back to the light energy. So while there are other sources of energy, the light energy is a primary source for everything going on Earth. Photosynthesis always begins when energy from light is absorbed by proteins that contain green chlorophyll pigments. So the autotrophs that perform photosynthesis don't always look green, but somewhere in their body, they're going to have these chlorophyll pigments that are able to take in and absorb light to start the process of photosynthesis. And photosynthesis occurs in two stages. You have your light reactions or light-dependent reactions, so these depend on light to happen, and it's when organisms capture and store the light energy. The light energy is converted to chemical energy in the form of NADPH and ATP. So in this light reaction or light-dependent reaction stage, the light energy is actually captured, turned into a chemical form of energy that can be stored or immediately used if necessary. In the carbon dioxide fixing reactions or light-independent reactions, these are ones that can happen even whenever light isn't present, as long as there was some energy harnessed in that plant already. So these don't have to happen while there is light present supplying energy. They can happen later. And this is when the formed NADPH and ATP, so it's already stored and formed in that autotroph, those two energy molecules drive the reduction of carbon dioxide to more useful organic compounds, such as glucose. And they are also referred to as the Calvin or Calvin-Benson cycle. So when the carbon dioxide is reduced, that process can be called the Calvin or Calvin-Benson cycle as well. So even though this is a light-independent reaction, it does still depend on molecules that were created by converting light energy into a stored chemical energy. So even though we call them light independent, they really couldn't occur if you didn't have the light transferring, the light energy that was able to be transferred to these energy molecules that are stored chemically. The main thing to remember about photosynthesis is that it is the process by which autotrophs can make their own energy using light. Plasma membrane. The plasma membrane is also known as the cell membrane or the cytoplasmic membrane. It separates the interior of the cell from the outside environment. So it being called the cell membrane makes sense because it surrounds the whole cell and keeps it safe from the outside environment. And the cytoplasmic membrane also makes sense because it's holding all the cytoplasm in. It's a membrane surrounding all the cytoplasm in the cell and protecting it from the outside environment. The plasma membrane controls the movement of substances into and out of the cell. So it controls what passes into the cell and what passes out of the cell. So if there are things that are waste and need to get out of the cell, the plasma membrane is controlling that. If the cell needs something, the plasma membrane is going to be what lets it in. And the plasma membrane is composed of a phospholipid bilayer and embedded proteins. Now, a phospholipid bilayer you may not have heard of before. So let's describe it. The phospholipid bilayer consists of many phospholipid molecules arranged into two oppositely facing layers. So this thing here would be your phospholipid bilayer. So bilayer because it's technically two layers of these phospholipids, and you can see how it kind of looks like it could be making a wall, and this whole thing would go all the way around a cell. Now, these top parts are phosphate groups, and these are fatty acid tails. So you've got phosphate group and fatty acid tails, which are like lipids. So you get your phospholipid bilayer because you have two layers of it. And these phosphate groups facing out bunch together and they repel some things. So they're going to repel what the plasma membrane wants repelled, and then they'll kind of open up and let through what the plasma membrane wants to let through. The plasma membrane also helps anchor the cytoskeleton to provide shape or structure to the cell. So the cell kind of looks like a big blob or a very small blob, and the cytoskeleton helps give it some shape, and the plasma membrane kind of holds that cytoskeleton in place because otherwise it doesn't have anything to anchor to hold on to keep that shape. The plasma membrane attaches to extracellular matrix and other cells to help group those cells together to form tissues. So, on the inside, the plasma membrane is holding onto the cytoskeleton and helping give the cell shape. On the outside, the cell membrane is reaching out to the extracellular matrix, to what's outside of that cell, and holding onto other cells to help group them into tissues. Because you usually don't see one cell by itself. Your body is made up of lots and lots of cells, and those cells group together to form tissues, and the tissues group together to form organs, and all together your organs form up your body and make you who you are. So the plasma membrane has a big role in that. Now, something to remember is that plants, bacteria, and fungi also have a cell wall, which goes outside of the plasma membrane. And it blocks out the passage of larger molecules. So the phospholipid bilayer is going to keep out teeny tiny little molecules, but this cell wall is going to keep larger molecules from even getting close to the cell. So you might consider like a piece of fabric the kind of plasma membrane. More stuff can get through it, it's more flexible, where maybe a piece of cardboard would work for the cell wall. It's more rigid, it doesn't bend as easily, not as much is going to get through. Something can get through the cell wall, just as something can get through the plasma membrane. But the purpose is to try to keep out anything that the cell doesn't need or want in it, and to let in what the cell needs. So, plasma membrane has a few main functions you should remember. It is protecting the interior of the cell from the outside of the cell and limiting what comes in and what goes out. It is helping give shape to the cell by anchoring the cytoskeleton, and it is going to join with other cells to form tissues because cells aren't usually going to be by themselves. So the plasma membrane is a pretty important part of the cell. It's what keeps each cell separate from the other. Proteins. Proteins are large biological molecules consisting of one or more chains of amino acids, which are known as polypeptides. So a polypeptide is an amino acid chain, not just an amino acid, but the whole chain of them connected. Now, proteins have different structures based on how many of them are present. So the primary structure of a protein is its amino acid sequence. So just the one chain, the one polypeptide chain, and what sequence those amino acids are taking. The secondary structure is the regularly repeating local structures stabilized by hydrogen bonds. Now this is whenever we've got a few proteins interacting together and the local structures or the regular structures that these proteins will form just all on their own without any help. And some examples would be the alpha helix, beta sheet, and turns. So those are just some different shapes that the proteins can take whenever there are a few of them joining together with hydrogen bonds. Now the tertiary structure is getting more complex. It's the overall shape of a single protein molecule. So enough proteins have joined together to make a whole molecule, and the tertiary structure is referring to the spatial relationship of secondary structures to one another. So you've got your alpha helices, your beta sheets, and your turns, and it's how they interact together and what kind of shape they all form whenever they form into a single molecule. And you've got proteins joining together and then more proteins with hydrogen bonds joining together into a larger molecule and what that molecule looks like spatially. The quaternary structure is going to be the structure formed by several protein molecules. So you can see how we just keep adding proteins here to get different structures. And the more that are added, the more different that these structures are going to look because they're getting more complex. So several protein molecules are joined together in the quaternary structure, and this is usually called a protein subunit. So it's no longer one molecule, it's multiple molecules. So it's a subunit which functions as a protein complex. So it's going to be more complex, it's going to be able to process more complex functions. And a lot of the time what you're going to see is the quaternary structure of a protein because you have lots of them together and they are drawn toward each other and then they bond. Now let's look at some of the functions of proteins. We have structural proteins, and these give stiffness and rigidity to biological components that would otherwise be more fluid. They wouldn't have a lot of shape to them. An example is keratin. Keratin is a protein found in our hair, nails, in birds' feathers, and in animals' hooves, and it gives them a hardness that they wouldn't otherwise have. It's not a part of your body like your skin or an organ, and you can feel they're a lot harder than your skin would be. Like your nail and your hair don't feel the same as your skin feels or as your internal organs would feel, and that's because they have this protein present that gives them more rigidity. So structural proteins are going to make things stiffer and give them a harder shape. Next we have enzymes. And enzymes catalyze chemical reactions. So there's a chemical reaction that needs to take place, but it won't be able to take place until this enzyme is present. When the enzyme is present, it catalyzes the chemical reaction and speeds it up and lets it occur. Next, you've got receptors. Proteins that function as receptors bind a signaling molecule to induce a biochemical response. So it receives that signaling molecule and then it binds it somewhere that will induce the biochemical response that needs to happen. Next, you have antibodies, which are also known as immunoglobulins, and these bind antigens and target them for destruction. So an antigen is a foreign body that comes into the cell and it's not welcome, it's seen as the threat, and so these antibodies or immunoglobulins are part of the immune system, and they bind to the antigens or that foreign body and they target them for destruction so that then they are destroyed and removed from the cell and the threat is eliminated. We also have motor proteins, and motor proteins generate the forces responsible for muscle contraction. So think about your motor functions, being able to move your muscles, being able to move around. All of those are able to happen because of these proteins, these motor proteins. So if you're able to move your arm back and forth, if you're able to close and open your hand, those are responsible, those are happening because of muscle contractions, and those muscle contractions are generated by motor proteins. If we didn't have those, we wouldn't be able to contract our muscles like we are. Next we have pump proteins. So these are proteins that act as a kind of pump, and they transport ions or small molecules across a membrane. So you've got a cell membrane or an intercellular membrane, and the proteins can act as a pump and push these small ions or molecules across the membrane. And that is their primary function as a pump protein, is just to transport these small molecules or ions across the membrane. Lastly, we've got switch proteins, and these act as an on-off switch based on the presence or absence of certain other molecules in the cell. So if a cell was waiting for a certain molecule to be present to do something, then the switch protein is going to act as an off switch and keep the cell from performing that function until the certain molecule it's waiting on is present. Once it's present, it flips the switch and says, basically, it's on. It can perform this function now. So it acts as like an on-off switch for the cell. As you can see, proteins are very important in a cell. They actually can make up a large percentage of the components of a cell. For instance, you could have a cell that was made up of maybe 3% DNA, which is really important. You have to have the DNA encoded correctly for everything else in the cell to function. But the protein could make up almost 50% of that cell's components. So while it isn't maybe as biologically important as that DNA is, it makes up a big part of that cell, and not much would be going on if it didn't have protein there as well, because protein has a lot of functions. So you can see that proteins are very important for cell life and lots of cell functions. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together, and adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels pair together and your two curved abbreviations pair together. So adenine with uracil and guanine with cytosine. Usually, RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side, kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So, in viruses, RNA can carry the genetic material, and a lot of times, if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code. Viruses. Viruses are tiny, non-living, biological infectious agents. So while they are not technically alive, they are biological agents, and they are infectious. They infect healthy cells. They are going to consist of either a DNA or RNA genome encased in a protein coat known as a capsid. So this capsid is protecting the viral genome until it can get where it needs to go to infect a host cell. And it can only reproduce, this is viruses, they can only reproduce by infecting and taking over a living host cell. So while they are non-living, they must take over a living host cell to be able to reproduce their viral DNA or RNA. Now, once they're inside a host cell, the virus may remain dormant for a while. So it doesn't always immediately enter the living host cell and attack. Sometimes it remains dormant for a while, but eventually it is stimulated into the active or lytic phase, and the virus inserts its own genetic material into that of the host. So the host cell is unaware of this, and the DNA that is, or RNA that is mixed in with the host DNA or RNA, it doesn't recognize that right away. So the virus is able to take control of the host's DNA by mixing with it and then taking over it. It causes the host cell to produce many viral genes and proteins. So once it gets in there, it starts saying reproduce, reproduce, make more genes and proteins. And the ones that it are making, the ones that this cell is making are going to be new viral proteins and genes instead of the normal cell's genes and proteins. And then all of these viral genes and proteins combine to form new virions or virus particles that destroy the host cell and are released to infect other cells. So what happens is, once the virus becomes active and it's in the lytic phase, it's going to insert its own genetic material into that of the host cell, so probably into the nucleus. The virus DNA or RNA is going to mix with the host DNA or RNA, take control of it, and then tell it to produce lots more of these viral genes and proteins, which combine to form new virions, and more are being made so fast that the cell can't keep up, and the cell is going to burst or lyse, which is where we get the lytic cycle or lytic phase from, because it is whenever the end result is going to be the cell lysing or bursting open. And once it bursts open, all these virions are released to infect other cells and go and repeat the process. Something to remember is that the genome of both DNA and RNA viruses can be either single or double-stranded. A lot of times a virus is going to not be too complex because the genome is only going to be 3 to 100 genes. It's not going to be very big, but it can be double-stranded if it is a more complex virus. So viruses are sneaky little things. They're going to be protected by their protein coat, the capsid, and insert their genome, their genetic material, into a host cell, take over, reproduce more viral genes and proteins, which form so many new virions that it causes the cell to burst open and send out more viruses into the host to attack other cells.
MCAT, organic chemistry. Catenation is the ability of carbon to form long chains. In fact, carbon atoms are unique because of catenation. They are unique among all of the other atoms found in nature. Now, carbon chains are formed because they form bonds, or tetravalent bonds, with other carbon atoms. So they form tetravalent bonds, which means that one carbon atom forms bonds with 4 other carbon atoms. Now, this structure can be repeated endlessly without disturbing the stability of the bonds or the compounds formed. And so they have a repeatable structure. In fact, chains can form branches, which can form sub-branches, which form rings and more rings. Now, carbon compounds can be split into two groups, with the first being open chain compounds or aliphatic compounds. And so these are organic compounds that form carbon-carbon chains, such as alkanes, alkenes, and alkynes. They are formed in animal or vegetable fats and do not have a strong aroma. Now, the second group is closed chain compounds or cyclic compounds. And these are organic compounds that form closed rings, such as benzene, vanillin, or phenol. Now, aromatic compounds are also cyclic compounds that contain a 6-carbon ring with alternating double and single bonds. Now, as their name suggests, they do give off a very strong aroma.
Today we're going to go over combustion and terms and concepts related to it. So if you look at the board behind me, we'll just start at the top here. When we talk about autoignition temperature, when we think about autoignition temperature, we're talking about the temperature at which a substance is so hot that it just bursts into flame without need for an ignition source. As things get hotter and hotter, most substances, though not necessarily all, have a point at which when they reach that temperature they will, if there is oxygen or other medium in which it can burn, burst into flame on its own without the need for an external source of combustion, heat, fire, anything like that. It just on its own, spontaneously. And so it would be tied into number 5 here, spontaneous combustion, no source of ignition. When we think about things that don't need a source of ignition and yet burst into flame, we can think in terms of maybe a compost pile. As the compost pile sits there, it's got hay and other things in it probably. It's strongly insulated, and there's fermentation going on inside of it through the bacteria, which is generating heat. And as the heat rises because of the insulation, there's nowhere for it to go. It eventually reaches a temperature at which ignition can take place without an outside source, and it bursts into flames. And so sometimes people do have compost piles that go up in flames because of the spontaneous combustion medium, the fermentation by the bacteria, raising the temperature, it's well insulated, and then boom, up it goes. When we think about combustion, we have certain types of reactions. Exothermic reactions are those that give off heat. They generate heat and force it out, sometimes light, sometimes noise in the form of explosions, but exothermics give off. Endothermic absorb or take in heat, and these kind of reactions. So there is a heat transfer taking place here. Exothermic forcing it out, endothermic pulling it in, absorbing heat inward. When we think of endothermic reactions here, you can think of these sports medicine instant cold packs that has a chemical reaction inside them and gets very cold very quickly. This is an endothermic reaction. So when we think about heat transfer, which is very much a part of combustion, we need to remember that what's going on is within the way physics works now, thermal equilibrium is always trying to be sought. So if there's something that's warmer next to something that's colder, the general motion will be that the warmer temperature will move towards the colder temperature until these reach equilibrium. If you think here of a rod that's been superheated on one end and is room temperature on the other end, what's going to happen then in conduction with this thermal transfer is the kinetic energy from the rapidly moving molecules and atoms down here on the hot end running into the colder ones, transferring their heat and energy that way. And so the heat moves down from the hot end towards the cold end, eventually reaching a place of equilibrium where this loses its heat, this gains the heat, and then they match. So heat transfer is very much a part of combustion. Conduction being one of those things, direct molecular transfer of heat until thermal equilibrium is reached. Another form of heat transfer is radiation. This is electromagnetic wave transfer of heat. Here we need to think in terms of the sun, which gives off electromagnetic waves or radiates radiation, and it heats the earth. It communicates it through the medium of a vacuum, space, but it still arrives here in the form of heat transfer, radiation, as it warms the Earth and strikes the Earth. So that would be a form of heat transfer radiation. And then convection. Convection is heat transfer through a fluid medium. Now when we say fluid, we don't automatically mean liquids. Fluids are also gases. So a fluid medium is a gas or a liquid. So you can get convection currents in air as well as in water. What you have then is heat is applied to the fluid, either gas or air, and as the fluid is heated, it gets less dense and warmer, and so it rises, causing the more dense, colder fluid then to move downward, and you get a circular motion. If we think in terms of boiling water, you can actually watch as it seems to churn up from the bottom. The heat source is there. The hot water becomes less dense, lighter, comes to the surface. The cold water sinks down, is heated, and comes up again. You can think in terms of air as well. Hot air, less dense, lighter, rises, cold air, more dense, heavier, sinks, is heated, and sent around. So you get these circular convection currents. So these are the main things to keep in mind around heat transfer: conduction, direct molecular radiation, waves, convection, this circular motion through a fluid medium, gas or liquid. We talked about spontaneous combustion in your compost pile under auto ignition temperature, and then we've got hypergolic reactions here, or combustion. Basically that's a substance when brought into contact with another substance that immediately ignites without an ignition source necessary. It's just that type of reaction, and this is what they do in rocket engines. They have two separate substances kept in different compartments, and then they are injected into a third compartment at the same time, and when they meet and react, you get combustion automatically without an ignition source, which then they use to propel the rocket upwards. So hypergolic substances are those that ignite or combust when brought into connection with an oxidizer. So this substance brought into an oxidizer immediately combusts, and of course you get rocket explosions directed beneath the rocket, which sends it up. So hypergolic combustion. Well, this has just been a brief overview then of things related to combustion, some of the concepts, some of the vocabulary in those concepts. If you'd like to learn more about this and related matters, if you'll look underneath this video, there's a link down there. And if you'll click on that link, it'll take you to the website that has that information. And while you're on that website, you'll also find a link to an ebook that's ready for immediate download. Today I want to go over briefly dehydration. Dehydration, causes, signs, best way to test for it, and best treatment options in terms of dehydration. I don't personally wish dehydration on anyone. I don't know if you've ever experienced it, but it is not pleasant. Last year, prior to Thanksgiving, I got violently ill and was unable to keep any fluids down of any kind. My body stopped taking in fluids. They went in, they immediately came back out. And eventually, because of the lack of intake of fluid, my kidneys shut down. I ended up in the hospital. It was very painful, not a pleasant experience. So dehydration is a bad thing. You don't want to experience it. But what we talk about dehydration, the main cause is, of course, lack of water intake, or excessive loss of water, or high solute load. In my case, I had both of those. Lack of fluid intake and an excessive loss of fluid because I was sick. And this can be especially dangerous in children. If they become violently ill and they have vomiting or diarrhea, you can lose a great deal of fluid in a very short amount of time. So anyway, dehydration, lack of water intake, excessive loss of water, it comes about due to sickness. It can come about because someone doesn't drink enough water and then they sweat a great deal, or because of their diet and other things, they have a high level of solute in their blood, a high level of things dissolved in it, various types, and this can cause dehydration as the fluids get all out of balance. Signs that someone has dehydration include fever, sweating, hyperventilation, rapid weight loss, decreased urine output, and in my case last year, zero output. The kidneys just shut down. Poor skin turgor. Skin turgor basically is if you pull up the skin on the back of your hand or behind the arm, you pull it up, tent it up, and let go. If you have good turgor, it goes back to its original place rapidly. If it goes back to its original position slowly, this is poor skin turgor. So dehydration, poor skin turgor tends to be a sign of that. And then increase in the solutes. Things like serum uric acid lab values go up and those sorts of things. Anyway, the best lab assessment for dehydration is to test for serum sodium. Now, normal level is 136 to 145. If it's outside of that, then you have a strong case for dehydration. The best treatment for dehydration, the best way to rehydrate the body is to take water orally by mouth or to put a 5% dextrose solution in the water. In my case, my body was unable to process it, and so it had to go in intravenously. But the best way to treat dehydration is drink lots of water. So this has just been a basic overview of dehydration, its causes, its signs, best lab assessment, best treatment. And if you'd like to learn more about this and related matters, underneath this video you'll find a link. If you'll click on that link, it'll take you to the website that has that information. And while you're on that website, you should also find a link to an ebook that's ready for immediate download.
Equilibrium is a constant state under a given set of conditions, but it can change to a different state if the conditions change. So notice here that equilibrium can change if the conditions change. And so that's why we call this dynamic equilibrium. Equilibrium systems are dynamic in that they can respond to stresses or changes in conditions. So if the equilibrium system is stressed or the conditions change, a dynamic equilibrium can respond. Or rather, I should say, an equilibrium can respond, and that's why we call it a dynamic equilibrium. Now, the French chemist Le Chatelier described how equilibrium responds to stress. So he came up with Le Chatelier's principle, which is if a system at equilibrium is stressed, the equilibrium will shift in the direction that relieves this stress. So an equilibrium wants to be in a situation of little stress. And so some common stresses include changes in, we're going to write changes up here, changes in concentrations, and this can be in the concentrations of reactants or products, changes in pressure. Now, changes in pressure are only going to occur in reactions where gases are involved. And then also changes in temperature. So if any of these things change, then we consider this a stress. And so equilibrium here, the equilibrium is stress, so the equilibrium will shift in the direction that relieves this stress so that it's in a situation of little stress. One final note is that catalysts do not change the values of a system in equilibrium. It only changes how fast the system reaches equilibrium. So again, equilibrium is a constant state under a given set of conditions, but can change to a different state if the conditions happen to change. MCAT, organic chemistry. A functional group is an atom or a group of atoms which defines the function or the mode of activity of a given carbon compound. And so again, a functional group is an atom or a group of atoms which is defining the function or the mode of activity of a given carbon compound. So here we have the name of a functional group, its structure, and then the corresponding class of compounds that it's in. So hydroxyl is one type of functional group with this structure, and its corresponding class of compounds is the alcohols. Then we have carbonyl with this structure, which goes along with ketones. Then we have carboxyl with this structure, which is in the carboxylic acids. And then amino with this setup right here, which is its corresponding class of compounds is the amines. And so these are all groups of atoms that are defining the function or the mode of activity of a given carbon compound. Hydrogen sulfide is made by acidifying iron sulfide. So you see right here, iron sulfide being acidified to produce hydrogen sulfide, which we have right here. Now, you may think that hydrogen sulfide is made by combining hydrogen gas and sulfur at high temperatures, but that doesn't work because hydrogen sulfide rapidly decomposes. And so, in the laboratory, a use for hydrogen sulfide is to analyze certain metal ions. Now, hydrogen sulfide can be produced a couple of ways in nature. One way is by volcanoes, and another is by the anaerobic decomposition of organic material. So it's the anaerobic decomposition of organic material by bacteria. So this could occur like in a swamp. Now, hydrogen sulfide is a poisonous gas, and it smells like rotten eggs. And certain bacteria found in volcanic vents on the seafloor use hydrogen sulfide in metabolism. So that's a couple of interesting facts about hydrogen sulfide. But remember that it's produced by acidifying iron sulfide and used in the laboratory to analyze certain metal anions.
Today we're going to go over 3 different forms of hypertension or high blood pressure. We'll begin with the first level, which is called essential hypertension. Essential hypertension is the most common form of hypertension. The cause of this type of hypertension is unknown. Most patients are also unaware that they have hypertension when they have this level, the essential form of hypertension. They don't even know that they're suffering from it. Usually it is only diagnosed in patients who have had 3 separate blood pressure readings of 140 over 90 or higher, and it's clear that those high ratings are not due to some other medical condition or disease. So normal reading, there's no other disease or medical reason. They have 3 separate occasions of a blood pressure reading of 140 over 90 or higher. They have essential hypertension. Most people are unaware of it until it has been diagnosed in this manner. Secondary hypertension is the next level. Secondary hypertension only affects about 5% of those who have hypertension. Once they've been diagnosed with it, it's usually due, the secondary form of hypertension is usually due to some other medical condition, such as renal disease, renal artery stenosis, aldosteronism, and pheochromocytoma. These are the sorts of things that would lead to secondary hypertension. Finally, the third level is malignant. Malignant hypertension is uncontrolled, severe hypertension and is life-threatening. It can be due to secondary causes, renal artery stenosis, or there may be no known cause. But the blood pressure levels are so high that they have to be brought down safely but rapidly. So the goal with malignant hypertension is to provide immediate treatment by decreasing the blood pressure over several hours. So no more than 25% of lowered blood pressure over a 1 to 2 hour period. This is usually done with medication, such things as nitroprussides, nitroglycerin, clonidine. Things like this are the most common medications used to treat malignant hypertension. So remember the 3 ones that we looked at today. Essential hypertension, this is the one that most people have. They don't even usually know that they have it, but on 3 separate occasions they have a blood pressure reading of 140 over 90 or higher that is not clearly due to another medical condition or disease. Finally, secondary hypertension, 5% of people have that, and it's usually due to another disease, renal artery stenosis, things like this. And then last is malignant hypertension, which is a life-threatening condition that needs to have immediate treatment by various medications where the blood pressure is brought down slowly, no more than 25% over a 1 to 2 hour period. If you'd like to know more about hypertension and related issues, please click on the link beneath this video. It'll take you to a website that'll have more information and an ebook that's available for immediate download. Today I just want to highlight some of the basics related to ligaments. So we're going to just go over what a ligament is, what it does, the functions it helps provide, just as an overview. So beginning with the fact that ligaments are dense bundles of parallel fibers that connect one bone to another. Ordinarily this would be in the joints. So where the bones come together in the joints, the ligaments would be part of the joint capsule. Although they are not always in the joint, they sometimes connect directly to the bones and other nearby bones as well. But ordinarily they are part of the joint. So dense bundles of parallel fibers that connect one bone to another. Now ligaments do not contract like muscles. They are not there to provide a movement or muscles. Their job is a passive strengthening and supporting of the joints. So ligaments are a passive way to strengthen and support joints. They do not contract nor provide the same sort of things that a muscle fiber would provide to the body. Ligaments also contain nerves, and these nerves are especially sensitive to position and speed of movement. Position and speed of movement. And we need this sort of feedback as we're moving around and doing various things. It's good to have that kind of neurological feedback. But along with that, it also protects us, because if the joints are moved too rapidly or in inappropriate angles or ways, it introduces pain. And this pain actually is helpful in preventing us from doing things that would seriously damage our bodies. So the pain is there to say, hey, don't go that direction, don't move that fast, don't do this with this particular joint. So the nerves are there to help us in positive ways, but also negatively to prevent serious injury. And ligaments are fully capable of rupture if they are placed under violent stress. So that's just been the basic overview of ligaments, these dense bundles of parallel fibers in the joints connecting one bone to another. They don't contract like muscles. It is a passive strengthening and supporting role in the body. They do contain nerves that are sensitive position and speed of movement, both providing a positive feedback for us and also negative feedback in the form of pain to prevent us from truly harming ourselves. And if enough stress is placed on ligaments, they can indeed be ruptured. Ask most snow skiers back in the day before they got good boot releases there, and they can all talk about having a tumble, ripping the ligaments in their knee, and needing some form of surgery. So anyway, that's the overview on ligaments. If you'd like to learn more, you can click on the link that's underneath this video. It'll take you to that website, and while at that website, you should find a link to an ebook that's ready for immediate download. The brain is one of the most important parts of our nervous system, and today we're going to go over the various parts of the brain and talk about some of the ways they contribute and help in the areas over which that part of our brain control. And so we're going to begin then with the frontal lobe. The frontal lobe controls our emotions, our judgments, and our motor aspects of our speech, and is also the primary motor cortex for voluntary muscle activation. After the frontal lobe, we have the parietal lobe, which receives sensory information from touch, proprioception, which has to do with our ability to tell where our arms and legs are and our orientation in space. So even with your eyes closed, you know where your arms and legs are. You have a sense of your body. This is proprioception. The parietal lobe helps govern that. Also our sensation of temperature and pain. That's the parietal lobe. Then the temporal lobe is responsible for our hearing, auditory information, and our language comprehension. The occipital lobe is our center for visual information, our sight. The cerebellum coordinates all of our muscle function. And then we have the brain stem, and within the brain stem we've got the midbrain, the pons, and the medulla. The brain stem governs our breathing, our respiration, our hearts, our cardiac center, and all the nerve pathways to the brain come through the brain stem. And then finally, moving on then from that to the diencephalon, which is the thalamus, subthalamus, and hypothalamus. The thalamus helps integrate and relay sensory information from the face, feelings there, our retinas, our cochlea, from our ears, our taste receptors. It interprets sensation of touch, pain, and temperature. So our thalamus is very important to not only our sense of what we feel in terms of pain, temperature, touch, but also our retinas, our eyes, our cochlea, and our ears, and our taste and our tongue. The hypothalamus controls the autonomic nervous system and the neuroendocrine system. It maintains our body homeostasis, helps regulate body temperature, helps regulate our appetites, governs our thirst and how we feel that, our sleeping cycle, and hormone secretions. So once again, the brain, very integral, necessary, core part of our nervous system, broken up into these various regions and what they controlled. If you'd like to find out more about the brain and its relation to our nervous system, I encourage you to go to the link that you'll find below this video. At that website, you'll find an ebook that is ready for immediate download. When we think about our pulse, it's something that many of us take for granted. We know we have one, we're alive, but we don't think about it much. And so we're going to go over a few things related to pulse today, and by answering a few questions. What is a pulse? Basically, when the left ventricular pumps blood into the arteries, their elasticity causes them to expand as they receive the oxygenated blood, and this expansion then is called pulse. The pulse should be rhythmic and known to be such, felt to be such, seen to be such. What is a normal pulse? Well, the average normal pulse is between 70 to 80 beats per minute. Now, there are things that can affect normal pulse. The average person has between 70 to 80 beats per minute, but beta blockers can affect a pulse. Someone with hypertension is going to have a higher pulse rate. Those with hypotension, or those who are in very good physical health, can have a lower heart rate. Some of them as low as 60 beats per minute can be common if they're exceptionally fit. A resting heart rate of 60 beats per minute. So we know what a pulse is as the left ventricular contracts and pushes blood out to the arteries expand, and it's this expansion as they receive the blood that's called the pulse as the heart keeps pumping the blood. We know what a normal pulse is. Now, where would you find the pulse? There are many places to check for a pulse, but the best place to look and sense a pulse is where an artery lies on top of a bone, so that you can then press on the artery over the bone and feel the blood as it's pumped by there. Basically, the palpitation of an artery over a bone is the best place to check, and the pulse is strongest and easiest to detect in the arteries that are closest to the heart, where the blood's coming out. Common pulse checkpoints then. We think first of all, very commonly, of the wrist. When taking a pulse, you need to use your index finger and middle finger, place it on the artery over the bone, and apply light pressure. You certainly don't want to apply so much pressure that you're actually stopping the blood flow, but light pressure there to feel for it. Never use your thumb to do this, because your own pulse rate in your thumb will interfere with feeling the pulse rate of the person you're trying to determine whether or not they have a pulse and at what rate their pulse rate is. So at the wrist, on the radial artery, is a common place where these fingers are placed and felt over the wrist bone. At the medial inside of the biceps, on the brachial artery, is a place that can be checked. And then one that we see a lot in TV and movies, of course, is on the neck, just outside the larynx at the common carotid artery. So again, the two fingers pressed there with pressure enough to feel the pulse, but not so much that you're cutting off blood supply. Then the way you determine someone's pulse rate is you don't have to feel it for a full 60 seconds. You basically hold your fingers there over the artery, counting their pulse for about 15 seconds, and then you multiply by 4, and this will give you their average pulse rate. So we've answered the question of what pulse is, what a normal pulse rate is, where and how you determine pulse, these common places of wrist, bicep, and neck, and then how long you need to actually feel someone's pulse in order to get their beats per minute. If this is something you'd like to learn more about, then please click on the link underneath this video. It'll take you to a website where you can get more information, and on that website you'll also find a link to an ebook that's ready for immediate download.
Amino acids consist of a central carbon atom bound to 4 groups. Now, in every amino acid, 3 of those groups are always going to be the same. They're going to be made up of hydrogen, amine, and a carboxylic acid. So it's the fourth group that is going to differ in the 20 naturally occurring amino acids. And so this fourth group could also be called the designated R. So R basically just stands for any type of chemical chain. And so I'm just going to write here, it differs. Because like I said, although 3 of the groups are going to be the same for every amino acid, the fourth group is going to differ from amino acid to amino acid. So we can classify amino acids into 5 different groups. And so we're going to take a look at those classifications. So the first is nonpolar, which is where the R's are mostly alkyl groups. And so that's like alanine, valine, and leucine. And then we have the polar group, which is where the R's are oxygen and hydrogen and a double bond between carbon and hydrogen, but they are not charged. So that would be something like serine, glutamine, or praline. And then we come to the aromatic amino acids, which is where the R's are rings. And so this is phenylalanine, tyrosine, those are some examples. And then another group is what we would call positively charged, or we could also call this basic. And by basic, I don't mean simple, I mean the opposite of acidic. And so here the R's are amino groups like lysine and arginine. And then last but not least, the fifth group is basically the opposite of this, so it's negatively charged, or we could call it the acidic group. So here the R's are carboxyl groups, and so an example of that is aspartate. So that's a look at the 5 different classifications of amino acids. When carbohydrates burn, they yield carbon and water. And so that's why the name carbohydrates comes from carbon and water. Now, carbohydrates include sugars and starches. And so sugars and starches are the main source of energy that your body uses. So you can see that carbohydrates are very important. Now, there's 3 basic types of carbohydrates I want to go over. First being monosaccharides. It's a really long word, has a complicated spelling with 2 C's in a row. But more important than the spelling is what a monosaccharide is. So it's a simple sugar such as glucose, fructose, and ribose. Now, we call these sugars monomers because these sugars are monomers for more complex ones. And so notice here in monosaccharides we have the prefix mono, which means 1. And then I just mentioned that these are monomers, which has that prefix again of mono meaning 1. Now you may be familiar with polymers, which has the prefix of poly, which means more than 1. So basically these sugars are monomers for more complex molecules. So these are single molecules that are the building blocks to be able to join together with other molecules to form polymers. So remember that because we're going to talk about that more in a little bit. So the second type of carbohydrate is a disaccharide, which is a double sugar such as sucrose, maltose, and lactose. Now, sucrose is what we would call table sugar. So it's the type of sugar you use for everyday use, for baking, coffee, that sort of thing. Now, the third type of carbohydrate is starches, which are large biological polymers that consist of straight or branched chains of monosaccharides linked together. So let's go ahead and break that down because that's kind of complicated. So these are large biological polymers. So we see that prefix of more than 1. And remember, I talked about these being monomers. They're going to join together to form the polymers. And so we see that played out right here because these monomers join together to form polymers. And we even see that confirmed or spoken of again later in this definition. These polymers consist of straight or branched chains of monosaccharides linked together. So starches are basically made up of monosaccharides linked together either in straight or branched chains. Now, in complex sugars, the monosaccharide monomers combine by condensation reactions where water is removed in the form of a hydrogen atom from one monosaccharide and a hydroxyl group from an adjacent monosaccharide. So that's a look at the 3 types of carbohydrates. The electromagnetic spectrum is defined by frequency and wavelength. Frequency is abbreviated as a lowercase f and is frequently measured in hertz. And wavelength is abbreviated as a symbol that looks kind of like a lowercase h with more squiggles. And wavelength is measured in meters. Now, because light travels at a fairly constant speed, frequency is inversely proportional to wavelength. This concept is illustrated in the formula frequency equals c divided by wavelength. And c stands for the speed of light. The speed of light is about 300 million meters per second. Now, frequency multiplied by wavelength equals the speed of the wave. And for electromagnetic waves, this is going to be the speed of light, with some variance for the medium in which it is traveling. Now, electromagnetic waves include many different types of waves, as you can see here. So what I've done here is I've arranged these different types of waves from largest to smallest in terms of wavelength. So we have radio waves, microwaves, infrared radiation, visible light, ultraviolet radiation, X-rays, and gamma rays. Now, the energy of electromagnetic waves is carried in packets that have a magnitude inversely proportional to the wavelength. Isotopes are atoms of the same element. They are similar but a little bit different. They have the same number of electrons and protons, so they have the same atomic number, but they have different numbers of neutrons, which gives them different atomic masses. So one way to show the different isotopes of an element is by giving the element name, putting a dash, and then giving the atomic mass. So for hydrogen 1, there is 1 proton, 1 electron, and 1 neutron. So the atomic number is 1 because the atomic number is the amount of protons in an atom of that element. So there's 1 proton, so the atomic number is 1, and then the atomic mass is 1 because we see that number right there. Now for hydrogen 2, there's 1 proton, 1 electron, and 2 neutrons. Notice the number of protons and electrons isn't changing. So the atomic number is still 1, and the atomic mass is 2. Now the third example we have is hydrogen 3. Again, there's 1 proton and 1 electron. That's not going to change, so the atomic number is still 1. And then the atomic mass is going to be 3. So that's 3 examples, different isotopes of the same element. Now, isotopes of any given element are going to behave the same chemically. This happens because chemical activity is determined by the number of valence electrons, not the number of neutrons. So all isotopes of a given element have the same number of valence electrons. So they behave the same chemically. Light is the portion of the electromagnetic spectrum that is visible because of its ability to stimulate the retina. Light is absorbed and emitted by electrons, atoms, and molecules that move from one energy level to another. So an atom might release energy to move to a lower energy level, but absorb energy to move to a higher energy level. Now, visible light is between ultraviolet and infrared light on the spectrum. So it's anywhere from 380 nanometers all the way up to 760 nanometers. So 380 would correspond to the color violet, and 760 would correspond to the color red. This is because the human brain interprets or perceives visible light as color. For example, when the entire wavelength reaches the retina, the brain perceives the color white. Now, when no part of the wavelength reaches the retina, the brain perceives the color black. Atoms and molecules can gain or lose energy only in particular discrete amounts. Therefore, they can absorb and emit light only at wavelengths that correspond to these amounts. So, using a process known as spectroscopy, these characteristic wavelengths can be used to identify substances. Some common types of lipids that you are probably familiar with are fats, oils, and waxes. Now, like carbohydrates, lipids are organic molecules. However, lipids have a more complex structure than carbohydrates and are larger molecules. Now, lipids include several different types of groups. And so the first is triglycerides. And so stated somewhat simply, triglycerides consist of a glycerol molecule that is bound to 3 long-chain fatty acids. So glycerol is a polyalcohol and fatty acids are long-chain carboxylic acids. So triglycerides are the most common lipids and make up fats and oils. Now, a second grouping is phospholipids. And these consist of 2 fatty acids bound to a phosphate head group. And so the fatty acid portions avoid water, meaning they're hydrophobic, while the phosphate head groups seek water, meaning they're hydrophilic. So phospholipids have a structural function. They make up a cell and organelle membranes in your body. A third group is steroids, which are lipids that are derived from cholesterol. Steroids have a 4-ring structure with a hydroxyl group at one end and a short carbon chain at the opposite end. Cholesterol is also found in cell membranes and circulates in the blood. Another type of steroids are hormones. Now, the fourth group of lipids that we're going to talk about are waxes, which are formed when a long-chain alcohol binds to a long-chain fatty acid. So again, some common types of lipids are fats, oils, and waxes. And like cholesterol, lipids are organic molecules. A molecule is an electrically neutral combination of 2 or more atoms of the same or different elements joined by covalent bonds. So I want to go ahead and break down this definition a little bit to make sure we really understand it. Notice up here it says that the molecule is electrically neutral. So that means it has no net charge. So any positive or negative charges in that molecule cancel each other out, so there's no net charge. In other words, it's neutral. Now, it's a combination of 2 or more atoms. Now, so this could look like this. 2 hydrogen atoms or 1 oxygen atom and 1 hydrogen atom together would work as well because they have to be of the same or different element. So either one of these works as a molecule. Now, these are some really simple examples of molecules. And so molecules can be simple like those right there, or they can be as complex as large biochemical macromolecules such as proteins, starches, and cellulose. Now, proteins can range in size from 50 to more than 2,000 amino acids linked by peptide bonds, while starches and cellulose molecules can consist of more than 10,000 smaller glucose molecules covalently linked together. Now, there are some substances out there, 3 in particular that I want to highlight, that you may think are molecules which actually are not classified as molecules. So I want to go over those. So one is aggregations of polar molecules joined temporarily by hydrogen bonds. Another example is electrically charged ions joined by ionic bonds. And then finally, metals consisting of positively charged ions in a sea of delocalized electrons. So these three substances that I just listed, you may be inclined to classify these as molecules, but they actually are not. So I just wanted to warn you about those. So let's just review the definition for molecules again. That's something that's electrically neutral of two or more atoms of the same or different elements, and they're joined together by covalent bonds. So remember all these things. They're neutral. There has to be two, but there can also be more. Same elements or different elements. And they have to be joined by covalent bonds. So generally, when you have a definition of something, it just usually is maybe twofold. Here, there's lots of different criteria something has to meet in order to be a molecule. So you want to be able to remember all these so that you can see a substance, learn a little bit about it, and be able to identify if it is indeed a molecule. Organic compounds are basically compounds that contain carbon. There are two main ways to classify organic compounds. The first is by classifying where they come from. So you would have natural organic compounds and synthetic organic compounds. The second classification system is based upon how large the compounds are. So you have small molecule and polymers. In this case, we're going to take a look at the first classification system. So natural compounds are just what the name implies, compounds that can be found naturally in nature. So these natural compounds are produced by plants and animals, and humans take those compounds and use them for their own use. Sugar is a great example of a natural compound because it's naturally occurring in nature because it's produced by plants. Synthetic compounds are produced by humans, and humans produce it and then use it for their own use. Now, sometimes these synthetic compounds can actually also be produced by nature, but for whatever reason, industry has found it more efficient to produce it synthetically. A great example is rubber. Rubber can be produced naturally by plants, but industry today finds it more efficient to synthetically produce rubber. So that's a quick look at organic compounds and how they can be divided up into natural organic compounds and synthetic organic compounds. pH is basically a measure of how many hydrogen ions are in a substance. pH is very important to many fields such as biology, chemistry, medicine, food science, farming, and water treatment. I've written up here on the board a pH scale ranging from numbers 0 to 14, each indicating the amount of pH in a substance. Now there are pHs that go higher than 14, but for practical purposes, this is the largest pH scale we need. Water has a pH of 7, and any substance with a pH larger than 7 is considered a base. So everything from 7 over to 14 is considered a base. Common bases that you probably know of are baking soda and bleach. Now any substance with a pH below 7 is considered an acid. Some of those that you may know of are lemon juice, tomato juice, and gastric acid. So water is kind of the middle bar on the pH scale, and anything to the right is the base, and anything to the left is an acid. MCAT, Organic Chemistry. We're going to take a look at 6 of the physical properties of alcohols, starting with physical state. So at normal room temperature, the physical state is going to be that of a liquid, and it's going to be a colorless liquid. Now, when it comes to odor, the lower members of the alcohol group are going to have a fruity smell. It's just a fact that the lower members of the alcohol group have a characteristic fruity smell. Now when it comes to density, alcohols are lighter than water. And alcohols such as methanol and ethanol are completely soluble in water. Now, on the other hand, the longer chain members of the alcohol group tend to be less soluble in water. Now, when it comes to acidic nature, alcohols are neutral liquids. So they have no effect on litmus or other acidity test. And then finally, conductivity. Alcohols are non-ionic and non-conductive. So the reason that alcohols are non-ionic and non-conductive is because alcohols are covalently bonded compounds. So that's a look at 6 of the physical properties of alcohols. MCAT, organic chemistry. We're going to take a look at 5 of the properties of organic acids, starting with physical state. So the first 3 organic acids are colorless liquids. Now, when you get to 4 to 9 carbon atoms, organic acids that have 4 to 9 carbon atoms, they're going to be colorless, oily liquids. So in addition to being colorless liquids, they're also going to be oily. Now, when you get to organic acids that have a higher number of carbon atoms, higher meaning more than 9 carbon atoms, they're going to be colorless, wax-like substances. Now, on the subject of odor, the first 3 organic acids have very strong smells, very pungent smells. Now, when we come to organic acids that have 4 to 9 carbon atoms, they're going to continue to have strong smells. This time it's going to kind of smell like goat butter. And then when you get to organic acids that have even higher amounts of carbon atoms, the organic acids are going to be odorless. Now, when it comes to solubility, the first 4 carbonic acids are soluble in water. Now, as the molecular weight of these organic acids increases, their solubility in water decreases. So eventually, when you get to organic acids that have 10 or more carbon atoms, they're going to be completely insoluble in water. Oh, by the way, I forgot to mention when we were talking about physical state, in all of these circumstances, no matter how many carbon atoms there are, these are all at room temperature. So the physical state of the first 3 organic acids is a colorless liquid when at room temperature. So that's just a brief point I felt like I should make. Now, when we get to number 4, the acidic nature, organic acids are weak acids. So organic acids are just weak acids, and so the acidic nature decreases as the homologous series increases, and the lower members readily turn blue litmus paper to red. Now, finally, on the subject of fatty acids, organic acids are fatty acids indeed. So larger organic acids that have 1 carboxyl group are also called, you ready for this, monocarboxylic acids. So I'm going to go ahead and spell that out, monocarboxylic acids. So most of these acids are produced by the hydrolysis of fats, and so because they're produced by the hydrolysis of fats, then we call them fatty acids. And so they have many industrial uses, especially in the soap and detergent industry. So that's a look at 5 of the physical properties of organic acids. A polymer is a large molecule which is composed of repeating units of smaller molecules that are chemically bound together. So it's a large molecule, and it's composed of repeating units of smaller molecules, and we can call these smaller molecules monomers. And so a monomer is basically the subunit or the building block for polymers, because notice here the prefix mono and the prefix poly. Poly means more than one, and mono means one. And so this is just one molecule, but when these one molecules join together in repeating units that are chemically bound together, they form a polymer. Now, the repeating unit may be made of 1 or more different monomers. So some polymers are made up of the same type of monomer, whereas others are made up of different types of monomers. So we have names for those different types of polymers. So the first being a homopolymer. And the second type being a copolymer. So homopolymers consist of only one type of monomer, whereas copolymers consist of more than one type of polymer. Now, one other fact about polymers may form straight or branched chains. So it just depends on the polymer as to whether or not they're forming straight or branched chains. Now, there are two types of reactions that are used to make polymers. So one is condensation, and the other is addition. So the way condensation works is it deals with the removal of water in the form of a hydrogen atom from one monomer and a hydroxyl group from an adjacent monomer. So a new bond forms between those monomers. Now addition works by the electrons of the double bond within a monomer being rearranged to form single bonds with other monomers. So basically you're altering the chemical makeup of these monomers to form new bonds between monomers. And so that's a look at polymers. I want to take a look at the properties of solutions and some different types of solutions. But first off, we need to define what a solution is. And it's a homogeneous mixture of two or more substances in a single phase. So by homogeneous, we mean the same. In other words, you don't have two substances somewhat mixed together. They are completely mixed together, forming one substance or one solution. And they're in a single phase. In other words, they're either all the components of the mixture are either in liquid, solid, or gas form. They're not in different forms. So every substance in the mixture is combined together to be the same in the same phase. And in the case of a solution, the particles are distributed uniformly throughout the volume, and the molecules do not react chemically with each other. So in any solution, you have two parts, the solvent and the solute. Now, like we said, a solution can be made up of two or more substances, but here we're just going to pretend that there's two substances. So we have the solvent and the solute. We have one solvent and one solute. So consider water and sugar. So the solvent here would be water, and the solute would be sugar. So the solvent is what is dissolving something, and the solute is what is being dissolved. So if you were to put sugar inside water, water would be the solvent because it would dissolve the sugar, and the sugar would be the solute because it's being dissolved. Now, like I said earlier, when you put sugar into water, the sugar is not going to stay in there. You may go to the bottom at first, but if you stir it around, eventually the water is going to break down those sugar, the little pieces of sugar, until the sugar is completely dissolved, and the sugar is going to be distributed uniformly throughout the volume. If you were to take a teaspoon of that water, there'd be a certain amount of sugar in that water. And if you were to take another teaspoon out of that water, there'd be the same amount of sugar in that section of the water because the particles are distributed uniformly throughout the volume. And now notice that the water was in liquid form and the sugar was in solid form. But notice that they're in a single phase now. The sugar has become liquid form. And then we said they form a homogeneous mixture. There's not some pieces of sugar and some water. The sugar has now been completely dissolved in the water, so now you just have sugar water as a whole. And then also, the water and sugar are not reacting chemically with each other. Now, there are three different kinds of solutions: gaseous solutions, liquid solutions, and solid solutions. And now, the name associated with each kind of solutions describes the phase of the solvent. So in the case of a gaseous solution, the solvent is in the gas phase. And in a liquid solution, the solvent is in the liquid phase. And then, I'm sure you're getting the picture, in solid solutions, the solvent is in the solid phase. And then the solute can be in any phase. So that's just three ways to classify a certain type of solution. But the important thing to remember, if you just take one thing away from this session, is that a homogeneous mixture of two or more substances in a single phase is what we call a solution. Sound is a pressure disturbance that moves through a medium in the form of mechanical waves. And these mechanical waves transfer energy from one particle to the next. So notice here that sound needs a medium to travel through. And so that can be something like air, water, or some other matter. But either way, sound has to have a medium to travel through. And then the reason it needs to travel through something is because it's transferring energy from one particle to the next. Now, another thing you need to know about sound is that sound is transferred through vibrations. Vibrations transfer energy to adjacent particles. Now, this may be a little bit contrary to what you thought about sound, because you may think that sound is the actual movement of particles over a great distance. But actually, the particles aren't moving. Instead, vibrations transfer energy from one particle to an adjacent particle. And that way, sound moves out through the waves into other areas. But actual matter is not moving with the sound. The sound is just moving from one particle to the next. And so sound is transferred through the movement of atomic particles. And so these can be atoms or molecules. Now, waves consist of two things. We have compressions and rarefaction. So a compression is when particles are forced together. And so the opposite of that is a rarefaction. So this is when particles move farther apart and their density decreases. So you have a compression with particles moving together and a rarefaction with those particles moving apart. So one compression and one rarefaction constitutes a sound wave. Now, waves of sound energy move outward in all directions from the source. So you have a source right here, a source of sound, and so you have waves moving out in all directions. So actually, if you were to look at what this would look like scientifically, you'd have all these waves coming off, but that takes a long time to draw. And so different sounds have different wavelengths. And then the last thing that you probably should know is that sound is a form of kinetic energy. Now, when I say it's a form of kinetic energy, that's as opposed to potential energy, which sound isn't going to be potential energy because it's not energy stored up. Sound is energy in motion, so we call it kinetic energy. Elemental sulfur comes from deposits in the ground. Now, sulfur has two physical properties that are used in the extraction process, and those are low melting point and low density. Now, the extraction process is called the Frasch process, and there's basically 7 steps to how it works, but the steps aren't very complicated, so we're going to go ahead and dive right in. So starting out, a well is drilled into a sulfur mineral deposit. So after that well is drilled, superheated water is pumped into the deposit. And so this superheated water is usually heated to around 170 degrees Celsius, which is about 340 degrees Fahrenheit. So that water is pumped down into the deposit. And as a result of that, the hot water melts the sulfur in the deposit. Now remember earlier I talked about 2 physical properties of sulfur that are useful in the extraction process. And the first was its low melting point. So you've already seen that come into play. The low melting point of sulfur allows for the water to melt that sulfur. Now remember that although this is melted, the sulfur is still down in that deposit, but we're trying to extract that sulfur. So in addition to superheated water being pumped into the well, compressed air is going to be pumped down into the well. And so because of that compressed air, the molten sulfur comes to the surface because all that air is forcing it to the surface. Now remember earlier I talked about the two physical properties. Well, the second one was low density. So you've also seen that physical property come into play. The low density of sulfur allows compressed air to be able to push it to the surface. Now that the molten sulfur is at the surface, it can cool and solidify. And so from there, the sulfur forms a rhombic crystal. And so a rhombic crystal has 8 molecules. So those are the 7 steps to the extraction of sulfur in what we call the Frasch process.
Today I wanted to go over some of the effects of starvation, short-term, prolonged, and then extended periods of starvation, and then the changes that you see physiologically. And we're going to begin by talking about some of the triggers to starvation, and then look at some of those results of short-term, prolonged, and extended periods. So beginning with the triggers of starvation, wounds and illness really lead to a stress response, and that produces a hypermetabolic state. So this obviously isn't the only cause of starvation, but the body responds to a wound or to an illness by creating stress. And in that stress response, a hypermetabolic state is brought about. The body enters a hypermetabolic state, which leads to an increase in the body's need for calories and protein, and yet at the same time, due to the wounds and illness, you get a decrease in the amount of intake of calories and proteins. So once again, the body responding to a wound or to an illness, this puts stress on the body. That stress response leads to a hypermetabolic state, meaning that the body sends out a signal saying we need to increase or uptake the caloric and the protein intake, and yet at the same time, due to the wounds and illness, the caloric and protein intake goes down. So the body says, I want more, less comes in. This produces at least some level of a state of starvation. Now, over the short run, if this is not corrected, the wound isn't cured, the illness isn't taken away, the protein and caloric intake isn't increased, over the short run of this state of starvation, what you get is an increase in the nitrogen in the urine, a rapid loss of muscle and weight, and a greater output of urine. So one of the short-term effects of short-term starvation is an increase, strong increase in the nitrogen in the urine, a strong increase in the output of urine, and rapid muscle and weight loss. If it's not corrected and there is a prolonged period where once again the body is still under stress, it is in this hypermetabolic state wanting an increase in calories, an increase in protein, and that is not met, then what you start getting then is, after it's been going on for a while, is the weight loss and the muscle loss actually goes down. It doesn't happen quite as rapidly in this prolonged state, and you get metabolic acidosis. You get metabolic acidosis even though the weight and muscle loss, or because of the weight and muscle loss, is not going down quite as rapidly. What you do get, though, is a sharp increase in the ammonia in the urine and a decrease in the nitrogen in the urine. As you noticed here, short-term starvation saw a sharp increase in the nitrogen in the urine. Here, the nitrogen in the urine goes down, but the ammonia in the urine goes up. And then finally, if the starvation is not dealt with and it goes into an extended period, beyond this prolonged period, it becomes premorbid, cachexia sets in, you get weight loss, the mid-arm muscle circumference decreases greatly here, there's a sharp increase in the creatinine height index and the urinary urea, and then a decrease in the serum albumin, transferrin, and lymphocytes. So you can see how things change the longer the starvation goes on. So just once again, just to recap, due to some sort of stress on the body, for example, a wound or an illness, the stress on the body, the response to that stress is to send the body into a hypermetabolic state where the body is calling for more protein, more calories. However, that need is not met. The intake goes down due to the illness or the wound. And over the short term, the nitrogen in the urine goes up, the output of urine increases, and there is rapid loss of muscle and weight. If it's not dealt with over a prolonged period, the muscle and weight loss decreases at this stage, but metabolic acidosis kicks in and the ammonia in the urine goes up while the nitrogen goes down. And then over the extended period, premorbidity sets in, cachexia, weight loss, mid-arm muscle circumference decrease, The creatinine height index goes up, as does the urinary urea, while the serum albumin, transferrin, and lymphocytes decrease. That's just a basic overview of starvation and its effects over 3 different time periods on the body, and what causes it, those sorts of things. If you'd like to learn more about this, study further, there's a link underneath this video. If you'll click on it, it'll take you to a website that has that information. And while you're at that website, you'll also find a link to an ebook that's ready for immediate download. Stress. It's something that every human being goes through at one point in their life or another, often multiple times, and sometimes over a prolonged period. The universal human response to stress has been dubbed the General Adaptation Syndrome, or GAS for short. It has 3 stages. The General Adaptation Syndrome, or the GAS response to stress, has 3 stages, and we're going to go over those now. The first stage is called stimulation. At this stage, the stress has been introduced to the person, and their body responds by the hypothalamus stimulating the adrenal medulla to release epinephrine or adrenaline. Epinephrine and adrenaline are the same thing depending on what part of the world you're in. They're going to favor one or the other. Epinephrine is the Greek-derived word for this hormone, and adrenaline is the Latin-derived word for this hormone, but they're the same thing. So the hypothalamus stimulates the adrenal medulla and releases epinephrine. The glucose level rises, the heart and respiration rates increase, and the individual becomes alarmed. We've all experienced this. Stress comes in, things heat up, adrenaline or epinephrine is released, heart rate goes up, respiration goes up, and the person is in a state of readiness, alarm, fight or flight in some ways. So the gas response then is managed by the adrenal and the pituitary gland. So the first level then is stimulation. If the stress is not resolved at this level, then the second stage that the General Adaptation Syndrome goes to is resistance. At this stage, the body resists the stress by returning respiration and heart rates to baseline. The hypothalamus commands the pituitary gland to release ACTH, a hormone that causes the adrenal cortex to release cortisol. Cortisol is a chemical that maintains high levels of blood glucose. So this is once again the body's attempt to deal with whatever is bringing about the stress. So stimulation puts a person in an alarmed or heightened state. It persists, resistance comes in, the body trying to bring things into line, into a normal state. Once again, the hypothalamus is critical to all this. In the initial stage, it stimulates the adrenal medulla to release epinephrine. In the next state, it commands the pituitary gland to release the hormone ACTH, which causes the adrenal cortex to release cortisol to keep the blood glucose levels high. Finally, if the stress is not resolved, the third stage for the general adaptation or GAS syndrome is exhaustion. Here, the pituitary and adrenal glands are utterly fatigued. The physiological processes start to break down. And it is this stage right here, this exhaustion stage, that if it is prolonged is most damaging to health. So stress management is extremely important. Stress comes into everyone's life. The universal response of every human being experiencing various levels of stress has been generalized as the GAS adaptation, or the general adaptation syndrome. It has 3 stages: stimulation, resistance, and then exhaustion, this stage being the most damaging to health. The hypothalamus involved in this. First stage, releasing epinephrine. Second stage, releasing ACTH in order to get cortisol to come out and raise the blood glucose levels. And then finally, when the pituitary and adrenal glands are utterly fatigued, everything begins to break down and the health begins to deteriorate. If you'd like to read more and learn more about stress and find out exploration more in this area, then please click on the link beneath this video. It'll take you to a website where you can study further, and you'll also find an ebook there ready for immediate download.
The body contains 2 varieties of sweat glands, eccrine and apocrine. Eccrine sweat glands are ring ducts that run from the innermost layers of the skin to the body's surface. Also, eccrine sweat glands secrete sweat to cool the body. Apocrine sweat glands are larger than eccrine sweat glands. Now with apocrine sweat glands, these glands emerge from hair follicles, under the armpits, areolae of the nipples, and around the genitals. Now, apocrine secretes sweat with oils and pheromones. Now, quickly about pheromones. Pheromones are chemicals that are detected by animals using their olfactory or smell sense. But going back to apocrine sweat glands, they are odorless sweat until it is exposed to the bacteria of the skin.
We want to briefly today go over the wall of the human eye. The human eye has a wall that's made up of 3 layers, an outer layer, a middle layer, and an inner layer. They each have a different name and a different function. We're going to go over those briefly. So we'll begin with the outer layer of the eye wall called the sclera. The sclera is here in the outer wall of the eye, and it is for protection. The cornea, the front of the eye, is actually attached to the anterior portion of the sclera, both top and bottom. I've only drawn in a representation of the top. And the cornea's job is to refract light that enters from the outside in order to put it through the lens and eventually put it onto the retina to allow vision. So the outer part of this 3-tiered system of the wall of the eye is called the sclera, and it's primarily for protection and anchors and holds the cornea. The middle layer of the eye wall is called the choroid. The choroid's job is basically to keep the inside of the eye dark. You're trying to receive light from the outside for vision. You can't have light coming in from all sides. It's got to come through a very narrow and small area, and so basically you've got a protective layer, a layer that creates darkness, and then you've got an inner layer, and this inner layer is called the retina. The retina, receiving the light coming in through the cornea, is responsible for communicating color and to the optic nerve, and therefore giving us vision. So within the retina you've got both rods and cones. The rod's job is to take low light, colorless environments and to help us see, and the cones are for our color vision. I happen to be red-green color deficient, so my cones are messed up and I have a very difficult time distinguishing red from green. Christmas tends to be a very boring holiday for me with all these different shades of brown, essentially. So I get asked all the time, how do you see colors? It's hard to describe. I do see colors, but I'm deficient in being able to distinguish reds from greens. They are very similar to me. But anyway, this has just been a basic overview of the eye wall and its 3 divisions, the outer, middle, and inner, sclera, choroid, and retina. If you'd like to learn more about this or related things, underneath this video you'll find a link. If you'll click on that link, it'll take you to that website that has that information, and while you're on that website you'll also find a link to an ebook that's ready for immediate download. Today in this video we want to go over transferrin. Transferrin is extremely important to our health as it binds iron in our system and transports it to the places in our body that need iron to produce red blood cells. If transferrin is not doing its job, or it's not present in the amount it needs to be, or it's doing its job too well, you can have one of 2 problems. Too little iron in the system leads to anemia. Too much iron in the system leads to problems on the other side with high iron concentration and blood poisoning, things like this. So transferrin is extremely important. It's a blood plasma glycoprotein, and its job, of course, is to find and bind free iron, and it binds very tightly, yet that bond is reversible as it then has bound the iron as a protein, transports it to the liver or to the bone marrow, and when it comes into contact with certain receptors on the surfaces of cells in that area, it then releases its iron. Transferrin is primarily produced in the liver. It's also produced by the brain, but its primary place of production is the liver. It binds iron and transports it to the liver, bone marrow, and other places like that. When it encounters one of these receptors on the surface of a cell, the iron is released and transported to the inside of the cell via a vesicle, also known as endocytosis. Transferrin saturation can be measured and determined. It's the total iron concentration divided by total iron binding capacity. Now, when we talk about iron and iron binding and releasing it, this really does have profound effects on people with renal disease. Those suffering from kidney problems need to have between a 20 and 50% saturation level, according to the Kidney Disease Outcomes Quality Initiative guidelines. So when you've got end-stage renal disease, you need to try to keep this right here, transferrin saturation, between 20 and 50%. So transferrin is very important to our health as it binds iron, takes it to the places where red blood cells can be made. Remember, it is a blood plasma glycoprotein. It binds iron, takes it to the places like the liver, bone marrow, where they find these cells that have receptors on the surface, where it then grabs the iron from the transfer and transports it into the cell and then processes it appropriately. Well, this has just been a basic overview of transferrin and things related to it, its importance to our health, things like that. If you'd like to learn more about this or related matters, underneath this video you'll see a link. If you'll click on that link, it'll take you to the website that has that information, and while you're on that website, you'll also find a link to an ebook that's ready for immediate download.